Good day, everyone. On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Eurofins DiscoverX, I'd like to welcome you to establishing qualified bioassays for checkpoint receptors to implement in QC lot release, case studies on PD-1 and SIRP-alpha. My name is Elizabeth Lamb, and I'm the host and technical director for today's event. Now, I would like to introduce our presenters for today. Our first presenter is Jane Lamerden, PhD, Director of R&D for Eurofins DiscoverX. Our second speaker is Laura McAleer, PhD, Senior Scientist, R&D, and Bioanalytical Technical Development, Sartorius Stedham BioOutsource Limited. And finally, our moderator for the Q&A portion of the webinar is Alpana Prasad, PhD, Product Manager with Eurofins DiscoverX. Now, I'll turn the webinar over to our first speaker. Welcome, Dr. Lamerden. The presenter ball is yours.
Thank you, Elizabeth, and thank you all of you for joining us today for this webinar, the shared webinar between our colleagues at Sartorius Stedham and Eurofins DiscoverX. So I'd like to begin by introducing Eurofins DiscoverX very briefly. We are a company that is primarily product-focused within the Eurofins family, supporting programs from research discovery through to lot release. Our products division is headquartered in Fremont, California, and our expertise is really focused on cell line engineering, characterization, and assay development, both for early stage assays as well as for bioassays. And we brought our technology expertise to bear on developing innovative assays to solve various bottlenecks that are encountered by groups in discovery and lot release. We have the advantage of having relationships at many top global pharma, biotech, and CROs. And importantly, our products have been implemented in discovery and the development of drugs, including many current marketed drugs. So our assays are built on a particular technology known as enzyme fragment complementation, or EFC. And our focus, again, as I mentioned, is on cell-based assays. So we use this particular technology to engineer a number of different target-specific assays using this fragmentation concept, which is shown here in this cartoon. So the reporter is fragmented into two pieces, what we call the enzyme donor and the enzyme acceptor. And typically we fuse the enzyme donor to one receptor of interest and the enzyme acceptor to another protein that relies on ligand-mediated recruitment of that protein. When the two proteins are brought into close proximity, they produce an active enzyme through complementation, and we're able to detect that active enzyme by the addition of enzyme substrate, which produces a luminescent signal. We have been able to adapt this technology to evaluation of numerous targets of interest for drug discovery. Just some examples shown here, including GPCRs, kinases, and interleukin receptors. And what we'll be discussing today is adaptation of this technology for the evaluation of immune checkpoint receptors. The field of immunotherapy has really exploded in the last decade, and it's based on the premise of targeting the patient's own immune system to kill the cancer that may be present. And it targets different branches of the immune system, either the innate or the adaptive immunity system. Over the past decade, the focus of most of the therapeutic modalities have been targeting adaptive immunity, in particular checkpoint inhibitors such as the anti-PD-1 or anti-PD-L1 axis that are targeting T cells. More recently, there's been an appreciation that the innate immune system is also an important element for immunotherapy as well. And here, what we'll be talking about is a particular immune receptor that's expressed on the surface of macrophages that is an emerging area for therapeutic intervention. And this is the SIRP-alpha CD47 axis. So today we will actually be talking about two different assays, one targeting the PD-1 and the other targeting SIRP-alpha. So we're starting here with an example of our PD-1 signaling assay to illustrate how we engineer assays for these particular targets. So for example, on the left here, we have a cartoon that shows the mechanism of action of PD-1 signaling, where PD-1, the C-terminal tail of the PD-1 receptor, which is expressed on the surface of the T cell, becomes phosphorylated upon ligand engagement with its ligand, PD-L1, presented on the surface of the tumor cell. Upon phosphorylation, this results in recruitment of SHP phosphatases, which then bind to and dephosphorylate the C-terminal tail, and this results in additional signaling through proximal signaling kinases. So we've built an assay that allows us to capture this very early event in the activation of the PD-1 signaling cascade. We've done this by building a co-culture assay that involves expressing PD-1 fused to our beta-gal fragment and co-expressing that with the SHP phosphatase fused to the other beta-gal fragment enzyme acceptor. When these are co-cultured with ligand-presenting cells, PD-L1 or PD-L2, we're able to see a gain of signal. By contrast, if you block this signaling by incubating with an anti-PD-1 or anti-PD-L1 molecule, then you see a loss of signal. So this is the assay concept. That is how we are approaching the two targets that we'll be discussing today. And now I will transfer the ball to Laura, who will talk about qualifying our PD-1 signaling bioassay.
Thanks, Jane. I will continue with a case study on the qualification of the PathHunter PD-1 signaling bioassay as fit-for-purpose implementation in QC lot release. Immunotherapy enhances the immune system's ability to recognize, target, and eliminate cancer cells wherever they are in the body, making it a potentially universal answer to cancer. It's an effective treatment for patients with certain types of cancer that are resistant to prior treatment with chemotherapy and radiation, and have also been approved for the first line of treatment in several cancers. Immuno-oncology treatments therefore represent the most promising of the cancer therapeutics. By harnessing the patient's own immune system to specific cancer cells, the hope is that the targeted treatment will be more effective while also reducing the side effects of current treatment options. There are a number of monoclonal antibodies targeting immune checkpoints currently on the market and require a host of assays for characterization. When a biosimilar manufacturer begins development, they are required to submit a suite of characterization data. As a company, we develop and pre-qualify these assays, removing the time constraints for our clients. Programmed cell death 1 is an immune checkpoint inhibitor and a cell surface receptor found predominantly on T cells. Under normal circumstances, the role is to promote tolerance and downregulate the immune system. PD-1 normally interacts with its two ligands, PD-L1 and PD-L2, both found on antigen-presenting cells and tumour cells. It was first identified to play a role in preventing autoimmune disease through two mechanisms. The first to promote apoptosis of self-reactive cells, and the second to reduce apoptosis in Treg cells. It was later identified to play a role in the clearance of abnormal cells. Many of the new therapies are targeting the PD-1 pathway to boost the immune response to cancer cells. Nivolumab is a fully humanized IgG4 monoclonal antibody which functions as a checkpoint inhibitor, blocking the interaction between PD-1 and its ligands, which would normally lead to the inhibition of T-cell activation. Therefore, nivolumab can be used to treat cancers as it allows the continued activation of T-cells which attack and kill cancer cells. Nivolumab was first approved for treatment of melanoma in 2015, and since then the drug has also been approved to treat other advanced cancers. Clinical trials mainly demonstrated a reduced risk of death, but also reduced the risk of tumour growth and metastasis. The DiscoverX PathHunter PD-1 signaling bioassay is a ready-to-use kit which may be used as part of a package characterizing anti-PD-1 molecule such as nivolumab. The kit provides all the components and makes for a simple-to-set-up bioassay. First, the Jurkat cells expressing the full-length engineered PD-1 are thawed in media and seeded in 96 well plates, which is then incubated at room temperature for 15 minutes before transfer to a humidified incubator overnight. Towards the end of the incubation, a dilution series of reference and test material is prepared and incubated with the Jurkat cells for an hour. During this period, the U2OS cells expressing PD-L1 are thawed, transferred to the cell plate containing the PD-1 JERCAT cell, and the nivolumab then incubated at an hour at room temperature. Following this, a two-step detection is performed. Detection reagent 1 is added to all wells on the plate and incubated for 30 minutes in the dark, before detection reagent 2 is added and incubated for a further 3 hours, also protected from light. The plate can then be read. The substrate hydrolyzed by the detection reagent releases a chemoluminescent signal. Where the nivolumab binds to PD-1, it blocks interaction with the U2OS cells expressing PD-L1 and no signal is detected. To begin establishing the assay parameters, the plate map was designed to incorporate a reference standard, 2 test materials prepared at the same concentration as the reference, and a research-grade anti-PD-1 provided in the kit. The research-grade anti-PD-1 would act as a positive control. All 96 wells were utilised with a 10-point dilution series running across the plate and a no antibody control which contained both cell lines. Additional controls were included for the first run only to assess the background signal, which may result from either cell line alone. This control would not be required in subsequent runs. The results from the first assay are demonstrated here. The fold increase or assay window magnitude was greater than 9. Both the test material A and B exhibited a full sigmoidal curve, returned accuracies within 10% of the nominal concentration. Test material A and B were almost identical to each other, demonstrating the repeatability of the assay. Test material C, the research-grade anti-PD-1, also returned a full sigmoidal curve and an equivalent fold increase as nivolumab, which demonstrated the assay had performed as expected. It returned a potency of 19.3%, however, this is not a concern as the antibody was only to act as a positive control for the assay and not a similarity sample. The only further development deemed necessary at this point was the optimization of the dilution series to better capture the upper asymptote and more points on the linear section. The optimization of the dilution series was performed in run 2. However, there appeared to be some separation at the upper asymptote of the different test materials. The assay was repeated to determine if there are any operator environmental issues apparent. The run resulted in the same separation at the upper asymptote, which suggested there may be an edge effect interfering with the results. Further runs assessing factors such as incubator positioning, plate map, and letting the plate settle prior to transfer to the incubator were performed, with no improvements of results. Moving to a more traditional bioassay approach using the inner 60 wells meant we could still assess the 2 test materials per run. The final assay design implemented was to use the inner 60 wells to assess 2 test materials against the reference standard. A 10-point dilution series would be applied across the plate in a variable plate map design, where the reference standard and test material positions differ from plate 1 to plate 2. It was anticipated that the variable plate map should reduce the potential for bias. The assay format was performed as a single accuracy assessment prior to the qualification of the method demonstrated here. The ICH-Q2 states the following characteristics should be assessed when validating an analytical method. At BioOutsource, we use these guidelines as basis for qualifying our off-the-shelf assays for biosimilar compatibility studies. Accuracy is assessed at known concentrations relative to the reference standard over a range of 50 to 200% starting concentrations. Intermediate precision is assessed at 50, 100, and 200% nominal concentration, and the specificity is usually assessed using an isotype control. This ensures a wide range can be tested in our assays and allows us to support clients from clone selection through definition of critical quality attributes from an innovator population, right to finished product compatibility studies and lot release. We set up the following qualification study to allow us to assess the major requirements of ICH-Q2. Over the course of the study, we used 5 different operators, 2 different lots of bioassay kits, and the assays were performed over 9 separate runs. This allows us to assess the accuracy, precision, and robustness of the assay across the full range. Specificity controls are of the same isotype of the molecule undergoing assessment, but not expected to bind either PD-1 or PD-L1. We also assessed the sensitivity of the assay using various stress samples which will be discussed in more detail later. The qualification was performed by 5 different operators using 2 different lots of bioassay kits. The assay performed well run to run with consistent fold increase and stability with the EC50 results. The representative data here shows the results of the samples at 100% and the specificity sample at the top, and the 50% and 200% samples below. The specificity sample failed to bind to PD1 or PDL1 on either cell line and resulted in a maximal response at all concentrations. Each of the test materials here returned values within 12% accuracy of their nominal concentration, and the dilution series provided an excellent fit, captures the upper and lower asymptotes well, with several points on the linear part of the curve. The data demonstrated that the inter-operator precision was very good when different operators used the same reagent. The magnitude of the response was 9.7 and 9.1, with the curve shapes being highly similar. In addition, the results generated using the different vials of cells thawed on different days also shows good correlation among the potency results, demonstrating a robust assay performance. The overall results for the accuracy of the assay are demonstrated here. The accuracy of the assay was determined to be within 14.5% across the range of 50 to 200, and 85% of the samples were accurate within 10% of the nominal concentration. The intermediate precision of the 50% sample was 2% CV, the 100% sample 7% CV, and 200 was also 2% CV. Incorporating the accuracy of the intermediate precision assessment demonstrated excellent dilutional linearity across the range of 50 to 200%, with an R-squared value of 0.9906. The results obtained indicate that the PathHunter PD1 bioassay would be suitable for use as lot release or stability studies due to the low variability in the relative potency results generated between different operators on different days. Stress samples were assessed in the PathHunter PD1 signaling bioassay to assess the assay sensitivity. Nivolumab was subjected to forced degradation. One sample was treated at 60 degrees in a water bath for 2 hours, and the other underwent several freeze-thaw cycles. It was frozen at -80 for up to 1 hour, then thawed at room temperature for approximately 45 minutes. This was repeated 5 times. Each of the samples were analysed by ion exchange chromatography to identify the structural differences. The samples would then be evaluated in the bioassay. Ion exchange chromatography is a technique for the detection and characterization of charge variants of biological molecules. The charge variant analysis is important in the characterization of biotherapeutics because it provides information about product quality and stability. Separation occurs on an ion exchange column, with acidic species being released first and basic species last. We utilize this method to assess the effects of the heat treatment or freeze-thaw samples on nivolumab. Untreated nivolumab is used as a reference. The main chromatographic peaks are present in all samples. The freeze-thaw samples are near identical to the reference peaks. However, both acidic and basic species are present in different quantities for the heat-treated nivolumab, as shown here in the chromatogram. Due to the samples being separated using cation exchange, the most acidic components are eluted first and the most basic components eluted last. Comparison of the chromatogram shows that the heat-treated sample shows small differences in the acidic and basic species. A structure-function study would be required to determine the biological impact of these differences. Initially, each sample was tested at 100% nominal concentration compared to the reference standard. The heat-treated sample returned a result of 78.4% and an accuracy of -21.6. This inaccuracy is out with the accuracy of the qualified method and therefore suggested sensitivity to the differences observed in the ion exchange chromatography. The freeze-thaw samples returned a result within 1.1% of the expected concentration, which also correlated to the results of the ion exchange chromatography, where no differences were observed. In order to confirm the sensitivity to the difference, the heat-treated sample was repeated using a different nominal concentration and would aim to demonstrate the dilutional linearity of the sensitivity of the method. The sample was assessed at 143%, which returned an inaccuracy of -23.5%. This was consistent with the heat-treated sample at 100% nominal concentration and further demonstrated the bioassay to be sensitive to small structural differences. So to conclude, the benefits of the thaw-and-go PathHunter PD-1 signaling bioassay are the assay was accurate and precise with excellent dilutional linearity. There's reduced variability and increased reproducibility between analysts and on different days. It can be used for QC lot release due to low variability, good accuracy and precision. It has rapid format and is less complicated when compared with traditional assays. There is no cell culture required, therefore no upfront characterization and cell culture routine development. The thaw-and-go kit means we don't have a lag time or assay day limitations. It's a versatile method and you can use any anti-PD-1 or anti-PD-L1 molecule with very little redevelopment required. The assay is sensitive to small differences in the molecule, and it allows us to support clients from clone selection through to lot release of their product. Thanks. I'll pass you back to Jane now.
Thank you, Laura. So I'll now shift gears and talk about the optimization qualification of our SIRP-alpha bioassay for therapeutics targeting the CD47 SIRP-alpha signaling axis. As we mentioned before, we're focused on developing MOA reflective assays, and the in vivo MOA of the SIRP-alpha assay is shown in the cartoon on this slide. So SIRP-alpha is an inhibitory receptor that's expressed on the surface of macrophages and dendritic cells, and it recognizes its ligand, CD47, which is presented on the surface of most cells that are expressed in the body. If CD47 is lacking, then SIRP-alpha is actually able to phagocytose that particular cell, and this is how red blood cells that are aging are removed from our bodies. Much like many cancers and other tumors have evolved the ability to overexpress CD47 and thus evade the immune system and not be phagocytosed. So if one were to use an anti-CD47 or SIRP-alpha FC as a way to block this interaction, this results in upregulation of turnover of the tumor by phagocytosis. And so this is an approach that is being used in the clinic now. And for groups that are developing these particular assays, an ideal MOA reflective cell-based assay would quantify that phagocytosis endpoint. But such assays are very challenging to implement in QC. So we have approached it taking, again, a different way of looking at the MOA rather than a phenotypic endpoint. Rather, we are focusing on the molecular MOA, which is shown here on the right. So again, similar to our PD-1 assay, the SIRP-alpha inhibitory receptor becomes phosphorylated upon ligand engagement, and that again results in recruitment of SHIP phosphatases, which ultimately result in inhibition of phagocytosis. So we built an assay, again, similar to our PD-1 assay, where we're measuring recruitment of the SHIP phosphatase to the C-terminal tail of SIRP-alpha. Again, using our beta-gal system, which we've already described to you, we've essentially built what we're a co-culture system where SIRP-alpha is fused. Our PK tag and our SHIP phosphatase is co-expressed with that. So moving forward, we have evaluated the agonist responses of this particular assay. First of all, using soluble ligand rather than a cell-presented ligand, you can see that you do get a nice response, although it is not a super robust assay window. If you present the ligand on the surface of the plate as a plate-bound ligand, it does improve the assay window to about 13-fold. And the cell-presented ligand produces the most optimal assay window, as you can see here, with a signal to background roughly 5-fold or a little 4-fold higher than what we see with soluble ligand. So this is the mode that we chose to move forward with a co-culture assay. So to validate that we actually can block this assay, we have tested this with commercial anti-CD47 antibody. And here on the left, we're showing that plate-bound CD47, which we know can stimulate the assay, is able to be blocked with an anti-CD47 antibody with a very robust assay window. But the window is significantly better if you are stimulating the cells with cell-presented CD47. And you can see that there's a significant improvement in the sensitivity to the antibody when you use a cell-presented ligand. Finally, we have clients who are interested in the other end of the axis and actually blocking SIRP-alpha as opposed to blocking CD47. And here we've used the ECD of SIRP-alpha fused to the NFC. And you can see that you are able to see inhibition of this axis using this reagent as well. So we have moved from developing a cell line that expresses SIRP-alpha and allows us to detect the signaling event to development of a ready-to-use assay format and a much easier method that can be transferred. So briefly, the bioassay method using the ready-to-use cells is shown here on this slide, where we're thawing and adding our CD47 and SIRP-alpha frozen to use ready-to-use cells and adding them to the assay plate, prepare our antibody dilutions and add those to the assay plate, and then incubate that mixture for 24 hours prior to addition of our detection reagent. So in total, this results in an assay time that's just over 24 hours. And what you can see here is that the shape of the curve and the IC50s are very similar between the continuous culture, which is shown on the left, and the cryopreserved or ready-to-use cells, the graph shown on the right. And what we see is very comparable performance both in terms of IC50 and signal to background with the ready-to-use format. So in order to demonstrate that we can actually produce multiple lots from this particular assay, we have done a study where we've evaluated multiple analysts and multiple lots. So shown in this graph are data from analyst 1 who developed the assay and generated 2 lots, lot A and lot B, where both SIRP-alpha and CD47 lots were produced in parallel. And so what you can see in the light blue is that the first lot, lot A, gives pretty comparable performance with a slight difference in IC50 for the very first experiment. SIRP-alpha lot B with CD47 lot B gives very, very tight IC50 data in the 4 different experiments in which it was tested. And then if we look at interchangeability of the ligand and the receptor by testing SIRP-alpha lot A with CD47 lot B, shown here with this green circle, you can see that the performance is very similar to the SIRP-alpha lot A performance. And similarly, if we use SIRP-alpha lot B with CD47 lot A, shown here with the orange circle, that data performs, we see very similar performance to what we see with lot B with lot B. So the ligand and the receptor are interchangeable. Importantly, if we have that protocol transferred to a second analyst who prepared their own independent lots, in this case lot C, you can see that lot C performed very similarly in 2 different experiments, while the same analyst also ran lot B and lot A and saw very comparable performance to that of lot C. So the primary difference that we saw in this set of experiments is that within analysts, we see very tight interlot and interrun reproducibility in IC50. And not shown here is signal to background, but signal to background is also very consistent. There's slightly more variability between analysts, but actually the difference shown here is well within the range that we typically see for cell-based assays, even for a single analyst. And ultimately, the question is whether this small amount of variability would actually impact relative potency measurements. Well, what we saw is that it does not. This is qualification data from our SIRP-alpha bioassay, where 2 different analysts were running samples ranging from 50% to 150%. And the data shown here indicate that the accuracy, precision, and linearity of the assay are quite good. What we're seeing is data from 3 different runs from 1 analyst and 1 run from a second analyst. And you can see that just looking at any of the different values here for these different potency measurements, there is no significant difference between analyst 1 and analyst 2. And we have an assay with excellent accuracy, intermediate precision of 9%, and excellent linearity as well. So we've also shown that the SIRP-alpha signaling assay is highly specific. The data here on the left is showing you antibodies to different checkpoint receptor ligands. So for example, anti-PVR, anti-CD80, and anti-PD-L1, all antibodies that are inactive in this assay, whereas the anti-CD47 antibody is quite active. Similarly, we've looked at specificity related to the IgG component that may be used for client antibodies in this assay. So here, human IgG2 and IgG4 are essentially inactive relative to the anti-CD47 antibody. Finally, we've demonstrated that the assay does respond and detect changes in the antibody through forced degradation. So the pink curve here corresponds to treatment of our anti-CD47 antibody at 65 degrees for 17 hours, and we are able to detect a significant shift, suggesting that the assay will be stability indicating as well. So I'll just close by commenting that we have developed an MOA-reflective bioassay for quantifying therapeutic agents targeting the CD47 SIRP-alpha axis. It is an MOA-reflective co-culture format. We have developed an assay-ready format as well that demonstrates excellent lot-to-lot reproducibility and utilizes an easy-to-use method that allows you to achieve results in about 24 hours. Our preliminary qualification with a commercial anti-CD47 antibody demonstrates excellent accuracy, intermediate precision, and minimal bias, as well as excellent dilutional linearity. And we've also demonstrated that the SIRP-alpha bioassay is highly specific and stability indicating. So I hope that you've seen through the two different presentations shown here that we have several assays in our immune checkpoint panel that are excellent MOA reflective bioassays. We have a menu of other checkpoint assays as well that are available on our website. There's a target that we did not discuss here that you would find of interest. And importantly, we have the ability to develop custom assays for you that use a similar MOA or even different types of readouts. So not only the custom assay development, but assay qualification is possible if you wish to pursue that with us. So I'd like to thank you for your time, and then I'll pause for any questions you may have.
Thank you very much. And now we will turn it over to our moderator for the Q&A for today, Alpana Prasad.
Thank you, Elizabeth. Thank you, Laura and Jane, for your excellent presentations. We will now go through some of the questions that have been submitted, and please continue to send your questions. In interest of time, if you're unable to address all your questions, we will get back to you after the webinar. So the very first question I have is for Jane. Have you looked at the Jurkat cells alone in the assay, since most of the Jurkats would express the receptor of interest? Jane?
For the PD-1 assay, it is true that PD-1 may be expressed in Jurkats, but it's not expressed at a very high level. And in both assays, the receptor, the only signal that would be attained in the assay is coming from the labeled receptors, which is those that are actually fused to the PK component of the beta-gal. So if you were to put native Jurkats into the assay, we would actually not see any response.
Thank you, Jane. The next question is for Laura. Has or could equivalent testing been done with this method? Laura?
Okay, thanks. For the development and the qualification of this method, we used parallel line analysis. The difficulty with equivalence testing, especially at this stage, would be insufficient data to properly implement the equivalence testing. Going forward, it would be possible with validation, stability, or lot release, but we would have to discuss this in advance.
Thanks, Laura. Next question is for Jane. What is the variability of the thaw-and-go cells, and are these cells easy to handle?
The cells are very easy to handle. It's much easier for both assay formats, the PD-1 and the SIRP-alpha, It's much easier to work with the thaw-and-use cells than to maintain cells in continuous culture at the appropriate cell density that's required for the optimal response in the assay. So we find that the thaw-and-use are actually much more straightforward for clients to work with. For clients who are accustomed to working with the two cell lines, the performance of the assay-ready cells is actually very comparable to someone very skilled with working with the cell lines. So the variability is actually quite low, and I think you saw from both Laura's and my presentation that the accuracy in the assay is on the order of, or I should say intermediate precision is on the order of 10%.
Thanks, Jane. Next question is for you, Laura. How do you replace a potency release assay with another more informative potency assay during the late phase development?
So you would need to set up an analytical method compatibility study. It's important to demonstrate that the compatibility either equivalent or better for the test method performance of a modified or new method. And you could support the data further with using an orthogonal approach to the testing of your product.
Okay, thank you, Laura. This question is for either Jane or Laura. How to deal with the hook effect in bioassay?
Yeah, sure. So I would just say often hook effects are a result of an excess of test material in the system. So my suggestion would just be to try reducing the starting concentration.
Okay, thank you, Laura. We will have time for one more question. So this would be for Jane. How do you define your critical reagents and how do you qualify them?
So critical reagents are any reagent that can change over time and that are essential to the performance of the assay. Obviously, with respect to the reagents we supply, the ready-to-use cells are considered a critical reagent. And the way that we approach providing cells that will perform similarly over time is that we establish acceptance criteria for new lots, where we are comparing the performance of the new lot to the old lot in that qualification study. So we try to set very stringent criteria so that there is no drift over time. And yes, we just basically perform a new qualification study with the cells as they become manufactured. I'm sure Laura has some additional comments there related to her critical reagents.
No, I think you've covered it, to be honest.
Thank you, Jane. Thank you, Laura. So we have additional questions which we will respond to you in this week after the webinar. With that, I'll pass you back to Elizabeth. Thanks, everyone.
Thank you very much, Alpana. Thank you again to our presenters for today, Dr. Jane Lamerden and Dr. Laura McAleer, to our moderator, Dr. Alpana Prasad, and we very much like to thank Eurofins DiscoverX for sponsoring today. Most of all, I'd like to thank all of you who came and spent this time with us. We know you're very busy, and we very much appreciate your willingness to come and listen. So on behalf of Eurofins DiscoverX and CHI, I'd like to thank you all for your attendance, and we hope to see you at future web symposia. Thank you so much, and have a great day.